Hi, everyone. My name is Keenan Hartert. I'm a biology professor at Minnesota State University. I study cancer research and bioeducation, hence why I utilize Prism quite a bit. In today's video, we're going to use a very new tool that I'm very excited about called hierarchical clustering analyses. So I've performed these analyses where you have columns and rows and values in between, where often I'll have patients versus genes in RNA, and I want to see like what values of genes are on the inside of that heat map grid. But more importantly, which patients behave the same genetically and which genes behave the same way genetically. So we're going to perform hierarchical clustering, visualize the cluster rate relationships. I'm going to kind of walk you through dendrograms, and I use them a lot in my research too. So it's a really good tool. We're going to uncover some natural groupings for what we find there. And what this is really going to help you out with is when you have a big matrix of data with rows and columns and values in between, not only can heat maps graph that for you, but if you want to know who's most closely related to who, Prism has that capability now. So we're going to load in some rows and columns. I'm going to use some Likert data in this case from 1 to 5 on a rating scale with different students that I had. And we'll set that up. We'll get some multivariable setups going and kind of check out some of the data analyses that you have at your hands once you actually use the clustering analysis. So we'll talk about Euclidean versus Pearson, distance matrices, cophonetic distance, the merge maps, some silhouette plots as well. So let's get to it. So in multivariable, I'm going to run this through. I'm going to go ahead and grab from my spreadsheet some data right here. In this spreadsheet right here, it already detects, hey, I've got labels over here. These are names of my students, as you can tell. It's got a theme. Each one of these is a Likert scale, and Prism has rightfully detected that these are continuous data in that case. In this case, I actually kind of told it to. So perfect. First thing, I'm just going to graph this and say, hey, like, here's a heat map, columns versus rows and multivariable. Let's go right here. I'm going to move my little thing there. So over in here, I'm going to say, yep, like these are my Likert values right here. I'm going to say like intro data, maybe. There we go. And in this case, these two will be a bit self-evident. So we'll go here, make sure that you can have everybody showing right there and say labels type. What were my row titles? Oh no, what were my variable values? And notice right here, we come up against one of our first things, but there's a trick. So go over here. In multivariable, you actually have to click up here at change data table format. You want to actually say, hey, I actually really do want row titles right here. And then if you want, because some of the formatting pieces are going to want the row titles, some are going to want them as a variable. I'll kind of talk you through that in a bit. Down here as well, you can say, hey, I want these to showcase which column header I had down here. And again, some of this might take some adjustment for moving the heat map around, making sure the titles are feeding in. Today's video is mostly about doing the clustering analysis and going from there. So that's what we're kind of going to roll with, even though you can have a nice little heat map like this, do your formatting like I like, and again, you can customize it. I love my Viridius color scheme, so I'm not going to change that. However, we want to start getting into which students rated the things most alike and which things were rated most alike. So in this case, we're going to do hierarchical clustering in analysis. I do not need to cluster name because it's not a continuous thing. Leave it in though. It's going to know that since it's label, leave it. It's going to know not to like put it in the heat map essentially, but it will factor in when we want to label things. So go ahead and keep that in. From this point, I want to do both rows and columns. If you wanted, you could do one at a time. That's fine. Row labels could be row number. But again, see right there, I'm going to say, hey, put the name as the row labels right there because the column already has a title. All right, options. I'm going to say Euclidean because this is more of a distance measure where Pearson is a correlation measure. It depends on the data you have. In this case, I'll go with Euclidean for distance because that's sort of the more default use of hierarchical clustering. Up here as well, you can say standardize the data to like sort of a true mean of 0 so that lower values than that mean are going to be negative and higher than usual values of that mean are going to be positive. In this case, though, I'm just going to go ahead and use my raw data. That's fine. If you have really bouncy data, like RNA-seq, for example, where like the numbers are up and down and some of the genes aren't relative, for example, you can make them relative using standardizer center right here. And it's going to make things really nice, and you won't end up with one of those heat maps that's like really splotchy in some areas. So looking pretty good here. Again, I want the linkage. That's my main thing. There's a lot of options right here. For outputs, yeah, let's go ahead and run a few of these through. So cophonetic, like we're going to see, is the height of these dendrograms, these like phylogenies we're about to build. The merge map is going to show how close would it take to make two of these categories into one. And there is a actual mathematical distance for that, and we can kind of take that look. Distance matrix, again, is more like how far on a number scale are these factors. So right here, for sure, click this. Say heat map. Give me a silhouette plot too, because what that's going to show is that what is the most healthy number of clusters for each one. It's a good little reference, and it'll actually lead us into our k-mean stuff that we'll do later. So I'm going to actually make this the default. You can. I'll probably come back and do some more though. All right, here we go. So right on here, you have your basic stuff for the tab results. You have a distance matrix. You now can see, like a correlation matrix in a lot of cases where we say like how similar are people to who? In this case, the lower the distance, the closer one of the students is, the gym leaders here, are to being towards that other one. So like Lieutenant Surge and Blue are 4 away in this case. Relatively, that's pretty good. And remember, in a correlation matrix, Lieutenant Surge is perfectly at the same distance as Lieutenant Surge, right? So you can see who's further away from each other, sort of with a raw value. The same is going to be said right here for all of our factors that we just looked at. You can start to see, what are the distances here? Cophonetic distance as well. How far away in a cluster are some of these things from more of a height perspective? So in that case, let's go ahead and look over here at what we're talking about. Now, you can load up the dendrogram for the rows, and you can now see right here which one of the students answered this more similarly to each other, and which one of the students were the most separated, which one of the students was the most isolated in terms of their answers. It's probably going to be Erica over here, right? See how her distance is very far from these other families, essentially, that are forming. I would say right here, there are essentially 2 main clusters. This one right here, and then these 3 right here, potentially. Because as you go further up the dendrogram, you are getting further away from the Euclidean distance of how similar your answers were, how similar the data was. So looking over here, you can see that Lieutenant Surge and Koga, they were the most similar. Excellent. Now, if we look over at the factors right here as well, you're going to see different relationships start forming. So who is the most closely related? It kind of makes sense that, well, let's see. Politics and video games are pretty close. Travel and school and learning were pretty close. But if money was ranked high, so was school and travel and learning. Arts and being social was pretty close. Products and social media being semi-close on the Euclidean distance is pretty neat as well. Now, the heat map function is going to put this all back together for you. So we're sort of back at where we started with the heat map, but in this case, now we have these wonderful dendrograms right here. We can visualize the entire thing like I like. I know, sorry, my little cell borders. So again, over here, just make sure that just get everybody in that party right there. Perfect. As you're going, that's nice. So I'm going to click right here. I want everybody labeled right here. Wonderful. So now I can actually see how this map is sort of forming right here, how a lot of the 5s in this region right here. So people that shared and were pretty close, like Blue, Misty, Lieutenant Surge, and Koga, they shared a lot of 5s in some of these, like food, media, coffee, social learning type stuff. You can see areas where people had strong agreements to disagree, sort of. How like the Gary, Giovanni, and Blaine cluster right here did not really like pets, politics, or video games. Not going to judge. But what you can use this for is this massive, excellent use of how to show, a ton of data in here. Really nice. Again, you can always do, like I say, I really like putting the value in the grid line if it's not a crazy, or in the actual cell if it's not crazy, but we'll skip that for now. So a silhouette plot. This is going to actually tell us for the rows. It's kind of saying that in a perfect world, these are 2 clusters for the rows. So if we look back here, it's basically saying this is a family and this is a large family. Oh, sorry. And similarly, I think you can see in the columns as well, family, family. And if we look at the columns for silhouette plot, oh, never mind, it actually wanted 4. My guess was off. So in the silhouette plot for the number of clusters, it liked it the most when there were 4 clusters. So actually, if I go back here, yeah, I guess I would say for columns, it's probably counting that as a column, that is a column, yep, that is a column, and that is a column. Perfect. And it's saying that that is like sort of best fit, essentially. And that's going to lead us into this k-means clustering that the next video will follow. But from here, again, you've got a great setup for graph. You can do your colors. Again, if people know what a dendrogram is, you don't necessarily need that right there. You could label it as a specific dendrogram. If I look at my results, and again, we've got some other great stuff. Now that you've seen the maps, what you have right here is merge maps for columns and rows. What this is saying is who would merge first if you started saying like, you got to combine, then you, then you, then you, then you. And it's a good measure, again, of that cophonetic distance because cophonetic is that height on the dendrograms. It's lower for ones that would naturally merge, right? Because as you started to like squish it up and say like, you have to fit, you have to fit, school and learning and travel are going to go together. Food and media would go together. That's cluster 1 is right there. See that? So after you make this as cluster 1, money would then join cluster 1 right there. And so on and so on and so forth, right? Very cool little tools right here. This is a new capability of Prism. It fits really well with the multivariable setup. Again, it takes some moves. Like I said, make sure that you have a title set up with your labels because some pieces use the labels, some use the title. Perfect. But otherwise, you are looking at making a lot of sense out of a lot of data. So excellent job. Wish you all the best with your analyses. Thanks for joining us today.